Thank you for the opportunity to speak about novel therapeutic approaches. Towards the end of the talk, I will use some of my career journey as examples, but I'll start off with speaking a little bit about the history of pharmaceutical industry, how we came to where we are, and talk about the therapy evolution, and then also talk about the future therapies that we can expect from pharmaceutical industry. And then at the end, I'll talk about pharmaceutical jobs as you, as scientists who are finishing your degrees, looking towards a career in the future. What kind of prospects do you see in the pharmaceutical industry, and what can you hope to do when you do join? So with that, I'll start this history of pharmaceutical company through pictures. And this is an example picture from 1900 of one of the parent companies of Novartis called Ciba, based in Basel. And as you can see, at that time, this was the first sort of pharmaceutical production. It's in small scale, it's chemistry, and before most of the manufacturers like Ciba were producing either textile dyes or something that were involving chemicals, but this was the first foray into actually manufacturing pharmaceuticals. So a lot of small scale processes that you can see there. In contrast, Sandoz, another parent company of Novartis, in 1917 establishes the first pharmaceutical unit, and previously also Sandoz was a chemicals manufacturer. But here you can see a little bit more into research, into looking at chemicals and how to produce them for pharmaceutical grade, for example. And that's about 100 years ago. And we've come a long way from there. In 1996, from the merger of Ciba Geigy and Sandoz came Novartis. And in 2002, Novartis first started the Novartis Institutes of Biomedical Research. And imagine Novartis is a Swiss company headquartered in Basel, where there were a lot of manufacturers, chemical manufacturers. But the place where the Novartis Institutes of Biomedical Research is located is headquartered in Cambridge, in Massachusetts. And that is strategically placed so that we can partner with some of the institutions there, for example, the Broad Institute at MIT or Harvard. And as you can see, the picture and the landscape has completely changed to small scale processes, to a lot of human interaction, because it then becomes a innovation driven industry. And it's less about manufacturing and more about discovery and development and innovation. So that's why the location of NIBR in Cambridge. Now, forward the clock a little bit more in 2021, and you see that Novartis is now a focused pharmaceutical company, at least the pharma division. We're looking at different therapeutic areas, and in the same view, you can see that our manufacturing processes have become highly automated. There is less handling of small-scale equipment or reagents, and it's basically operators who are doing programming of large-scale fermentation processes with microscopic injection of fluids into very measured areas where you can look at monitoring the growth of these cells to produce proteins of interest. So it's a big evolution of pharmaceutical industry, historically from chemical manufacturers to now discovery and development focused, understanding human physiology and coming up with new therapies. And alongside the evolution of pharmaceutical company, you also see the evolution of treatments. And I want to show to you where we are at the juncture of opening up our exploration into new discoveries and new treatments. So what we consider as a conventional treatment today are, for example, molecular compound. And this you may see as a kind of a colorless powder or a colorless liquid, which you may inject or take once daily. And this is a chronic therapy that may last either as long as the symptoms stay or as long as the disease stays with you. And most of the effect of this is to ameliorate the symptoms for treatment. So as long as the symptoms last, you're taking these treatments. And the whole healthcare system is designed around these kind of chronic treatments that you are taking on a daily basis for a prolonged period of time. And now if you look at where we are now into future therapies, or at least currently, already we have many approved therapies, example, cell and gene therapy. Here, the raw material is not anything foreign. Often the raw material is the patient's own DNA or the cells, or a correction of these processes by putting them back into the patient. And often these are one-time treatments. And we have learned to do this through the discovery of the human genome, understanding protein pathways, over 20,000 genes being expressed, leading to multiple proteins. but we're at a place where we can interrupt and reprogram some of these processes, thereby changing the disease for a long-term course. And this offers a promise of potentially curing or correcting these pathways as well. And one of the challenges we face today is that the whole medical system, including the healthcare system and payers, is not designed for such therapies where it's a one-time treatment that can offer a cure. So we're at a point where we need to evolve, not just as a manufacturing and research organization, but as healthcare systems as well, to understand how to work with these new treatments. Now, I give you an example a little bit more on what constitutes a gene therapy, right? And on the left-hand side here, you see a gene therapy is a delivery of a gene directly to the patient at the place of interest. So, for example, if there is a dysfunction in a gene that is expressed in the neurons, is there a way to deliver the genes directly to the spinal cord? Or if there is a gene dysfunction in the eyes, for example, we can deliver the genes directly into the retina or into the ocular space so that you target those cells where you can correct the gene that is of interest to you. And this is done by using a gene combined with the viral vector, and the viral vector enables a entry into the cell all the way into the nucleus where the gene or the protein of interest can be manufactured. And this is a very beautiful process today that especially in cells that are stable, that are not regenerating constantly, like in the nervous system, where you can deliver these genes and you can constitutively express these proteins that can change the outcome for the patient. Another example where it's a little more complex is cell therapies, and this is used in cases of cancers. For example, if you have a B-cell associated leukemia, you can harvest some of these effector cells, like the T cells, from the same patient, and you can program those T cells to find the cancerous cells and to sort of engulf them or to lyse those cells. And you can re-inject these T cells back into the circulation to go and target the cancer of interest. And this reprogramming of the T cells, or often called as CAR-T, is done through a lentivirus approach, which is another virus, just like the adeno-associated virus on the left-hand side. Or you can use more modern technology like CRISPR while using some of these molecular scissors to directly delete or insert new genes into the T cells that can then go back into the body. So as you see, these are very complex processes, and this is not a manufacturing that you can take into a large-scale production plant. You have to harvest these cells, put them into a very high-quality machine to really grow them, to transfect them, and to put them back into the humans. So we're grappling with these new technologies and how we can manufacture these, but at the same time also working with the healthcare systems to deliver this. So looking into the future, what can we expect, right? So what we think, and this is not a stable picture, we think there are 5 dominant platforms, and there can be many more, but there's an interesting way to group them that is appealing to me. And the first of these is chemistry and chemical biology. And this is very similar to what we have as small molecules or new chemical entities that constitute drugs, but going much more than just as enzymatic processes, looking into either providing a substrate for the enzyme through a small chemical molecule. We're looking at more complex processes where you can work with protein formations or deformations and glue the protein into a certain state. You know, proteins often go between one or more states, and one of those states could be active and the other inactive. So if you can glue them in a certain state of interest using new chemical entities, that is a novel pathway. So this is possible now, or enabled us to look at enzymes or targets that were previously considered undruggable. And by understanding the conformation of these and finding those chemical molecules that can intercalate in the right area, you can then make them more druggable targets and develop therapeutics that can lock proteins into a certain state. A very sophisticated mechanism that goes into structural biology and discovery mechanisms. And another one, which is very common, this platform is over the last 20 years has fully blossomed, is biotherapeutics. And this includes natural substances from the body like amino acids, proteins, DNA material, RNA. All of these can constitute biotherapeutics. And the most common form you see today is antibodies. And this is the technology that we have honed very nicely over the last couple of decades, where if you have a certain cytokine or if you have a certain receptor that you would like to block, that you would like to soak up, you can use the body's own immune system to generate antibodies to that. And you can synthetically manufacture these antibodies and provide them to the patient of interest, right? And that's a beautiful way to target these receptors and cytokines that can give you a chronic treatment that is more complex to manufacture than chemical entities, but at the same time, safer, less toxicity, and more human, less foreign. So the degradation of these doesn't involve the liver or an excretion through the kidneys that doesn't harm any of these organs. Another, which we just talked about, is cell therapy. And you've seen a kind of an explosion of companies that are very interested in cell therapies, because by using CRISPR technology, by using delivery of genes using viral vectors, you have an ability now to reprogram cells that can go back into circulation and target cancers, can go back and produce genes of interest that is very important for you. And this is given a very powerful tool that can be potentially curative for certain cancers. So cell therapy is another platform, very difficult to commercialize, very difficult to produce. The process is more of a two-way process, harvesting of cells and then putting back of the cells, and manufacturing, which involves reprogramming these cells. Very complex, but at the same time, extremely attractive from a scientific perspective. And the third one we also talked about a little bit is viral gene therapy. And this is at a point where I think this can be considered, especially in diseases that are monogenic, where a single gene is involved, either through a dysfunction or a mutation, and you can replace or reprogram that particular gene in certain tissues. This is extremely attractive way of reprogramming them in order to potentially deliver curative therapies. And we have done that through the spinal muscular atrophy condition, where delivering this SMN gene to the neurons has enabled many of these little children to have a life expectancy that was much different from before gene therapy was available. And the final one, which is a little more complex, that involves some of these previous treatments identifying, is radioligand therapy. Here it is that you can, by combining a specific targeted approach to take an antibody or a protein of interest to the cancer, and combining that with a radiotherapeutic, you can provide focused radiation to the cells of interest by destroying cancer tissues. It's a very creative method where, by using small radioisotopes bound to certain proteins, that can be delivered to the target cancerous cells, and then preventing the rest of the body from being exposed to radiation. This is also being developed in Novartis, and many other companies are looking into this. And this platform of 5 different therapies actually opens up a box of new treatments that you can you can envision multiple diseases being targeted with this, especially that are not symptomatic, where you'd like a change in course of the disease over the long term and a promise of cure. Now, one example on this, and this is particularly a pet project of mine since I am overseeing some of the work now, is how can you use these novel therapies in battling the current pandemic that we have? And Novartis has partnered with a company called Molecular Partners, and we're using a technology called DARPin. And DARPin stands for a designed anchor and repeat protein. And we're using this to see if we can stop the proliferation of these different variants and find a stable protein that can bind to all of these variants of SARS-CoV-2. So how do we do it? On the left-hand side, you see an antibody, which is a fairly large structure, but the binding site is on these tip of the Y-shaped, right, the dark green here. And what is a DARPin is much smaller. It's a synthetic protein consisting of human genetic material and a bed of alpha helices with a very small binding pocket. So you can create a library of DARPins. And by stringing some of these together, you can create multiple binding sites. So what Molecular Partners have done is identified a triple DARPin that can bind to 3 different sites in the spike protein of SARS-CoV-2, and thereby sort of insulating it from multiple mutations and makes it more difficult to escape the DARPin. And this one is in clinical trials, and if it is successful, offers a much more scalable and an attractive method to look at these new variants, where we're having challenges today with some of these variants escaping the antibodies that were discovered only about 6 months ago and have been used in treatment. So another attractive approach, and something in the future to look forward to. I'll close my talk with what do you have to look forward to? What do you, as new scientists coming forward into life science industry, have to look forward to in pharmaceuticals? So my own background, I started off as a mechanical engineer that I earned in India, and I went into the United States to study biomedical engineering. And this is about 20 years ago. My first job was in consulting, and my first job in pharmaceutical companies was to look at R&D financial investments. And R&D financial investments for pharma companies is huge. We, as a percentage of top line, are one of the highest investors into research and development. And it's a very risky industry. So I was able to learn some of the instruments of how we do these analysis of what will happen 10 years from now. How does healthcare change? And what products that are being discovered today can be used 10 years from now into the market, for example. And we call these as target product profiles. And this becomes then a library of, what you call as a draft product profile that you then develop your science and clinical programs to identify if you can find a product that fits this profile. This was extremely exciting for me, and from there I've gone into development, and now currently I'm a project leader. So what I want to say from here is that there is no one way to do a career plan in a pharmaceutical company. It is an industry that is very risky for development perspective, and that's why we make so many programs and many of them fail. Although the risk profile is changing, this is still a risky industry, and that's why there is a big innovation premium. So the final takeaway messages for you is it's a very innovation-driven industry that provides an opportunity to extend your learning over the many years, to go through different stages. It's now at a point where data science being coupled with life sciences is really opening up the box to new discoveries and completely new opportunities like the digital therapies that we just saw in the previous session. And one can construct a long-term view in career in a pharmaceutical company by looking at different interfaces of functions. You can continue your learning journey going from a research scientist to a development, and especially at this time, I would say it provides a very attractive opportunity to also get into the ethics and equitable development, equitable delivery of pharmaceuticals. So I welcome you to engage with us here in the discussions, and I'll hand over to the next speaker now.